Christian, how did you get involved with stem cells?
I think it's a nice story of what I call the traditional scientific investigation.
Hmm.
You observe something, you don't have an understanding for it, you put forth hypotheses, you go and you test it and you move forward. And that kind of investigation took me from my background, which is brain research,
Hmm.
and basically shifting into stem cells. And it's when I was working with a product that you may know, Climath Lake blue-green algae, known as AFA on the marketplace, which stands for Aphanisum and Amphlus aqua, its species name. And so I was hired in 1995 to study that plant and provide and document the mechanism of action behind its effect on the immune system, on inflammation, and on the mind. I wouldn't say most, let's say 50% of consumers would report an improvement of mental clarity, a sense of well-being,
Mm.
mental energy. And then we identified very quickly that it's a source of phenylethylamine, known as the molecule of love, the molecule of joy. It's associated with affective behavior, attention deficit, or the reverse, focus. We documented that it contains phycocyanin, which is a COX-2 inhibitor, the polysaccharide supports the immune system. So we documented that fairly quickly. We published all of that. But I came across in this work, I came across people who reversed multiple sclerosis, liver failure, heart disease, emphysema, Parkinson, um, severe joint issue. And for me, the case that was, because you hear these stories and they're just stories. What do you do with them? From a scientific standpoint, they're just stories. And I have no proof that the story that they're telling me is not true. Now, why would they tell me that story if it's not true? I believe them, but scientifically, it doesn't have a deep meaning. But one day I received this package and it was a photo album. There was a letter with it. And the lady was describing how when she was 12, she was walking with a kerosene lamp. She tripped and burned herself on the face, chest, and arm, third degree. So she was heavily scarred all her life. And at 60 years old, somebody provides her with some AFA. She starts taking it. She says the scars started to inflame, get itchy, red, but she felt that it was healing itchiness. So she kept taking the product. She took a picture of herself in the mirror every week, and she says, go and look at the photo album. And 1 year later, there's no scars left. That I can't deny. So it started to kind of really stick with me, like, well, what is this product doing, touching the skin, the heart, the lung, the liver, the pancreas? And the whole shift in stem cell research is when I came across an article. The title was Turning Blood into Brain. It was, to my knowledge, we need to go back in 2001. Stem cells are only blood precursors, and everything we learned in neurology is that the brain does not repair. And that study described a stem cell from the bone marrow going to the brain and becoming a brain cell. So the thought was, if stem cells can become brain cell, and there were already in the literature 2 articles that I knew of describing stem cells becoming heart and liver, then what could it be in the body that makes a stem cells to become liver, brain, heart, but not the rest? Makes no sense. So that means it's just a matter of time. Scientists will document that stem cells can also become lung, pancreas, skin, and the rest. And if they can, then that means they are the repair system of the body. How do you want a stem cell to go to the brain, become a brain cell, and it's not its function? So we published an article in the journal Medical Hypotheses suggesting stem cells are the repair system of the body. And in the back of my mind, the question was, hypotheses, what if AFA is a stem cell mobilizer, but it was not a thing before, so nobody ever looked at that. So we acquired a flow cytometer that allows us to count stem cells and started to take our blood samples, count stem cells, take AFA, count a few hours later.
How does a flow cytometer function, or what is
A
that
flow
tool?
cytometer is a pretty sophisticated device, machine, but the concept is fairly simple. So it basically aligns all the cell one behind the other in a very, very fine tube. And at some point, the cell passes in front of a bunch of lasers, depending on this, how sophisticated your flow cytometer is, and a photoreceptor on the other side. And it's going to measure all the light emission that is coming from that cell, which means every single type of immune cells will reflect light in a different way. So you can identify them that way. Plus their size, because it can measure the shadow of that cell. So it gives you the size, it gives you the luminescence of the cell. But you can also then start to use markers like antibodies that are attached to, let's say, a red dye. and it's an antibody for a marker, a protein specific to, let's say in our case, stem cells, CD34. So you incubate all your cells with a bunch of markers. Let's say a stem cells does not have CD45 but has CD34. So you use markers of both, and then you will see if a cell passes through, does not have one of the marker that is not for stem cells but has the one for stem cells, then it's counted as a stem cell.
Excellent.
And you can, using this machine, you can count the number of cells having specific markers. So we can, and over the years, that's what we have done. You can count stem cells, but you can count small stem cells, V cells. You can count endothelial progenitor cells. You can count progenitor cells, meaning older stem cells that have started to commit into becoming tissues, those that are more primitive. You can start to document all of that with that kind of machine. So that's what a flow cytometer is.
Incredible. Thank you for explaining that.
And the study is very simple. So we do this crossover, placebo-controlled, double-blind. So the patient comes on one day, rests for about an hour, because your stem cell population is extremely small. So if you sneeze, for example, and you squeeze your liver, you have put in your circulation a lot of you could have put a lot of lymphocytes. It doesn't change how many stem cells you have, but it changes how many of them are in a, let's say, 1 milliliter sample, if you want, of blood that you want to quantify. So in a flow cytometer, when we do a study, we're limited as to how many cells we can count. It's a matter of time. There's a point your cells start to deteriorate. So we count 200,000 cells, but typically in the blood, out of these, you only have 200 stem cells. So you see, it's like you need to have a protocol that is really, really well designed and well normalized for all variables. So anyway, the person comes in the morning, sits for about an hour, rests. Then we take a blood sample. Then we give them placebo or a test product. And then we take another blood sample an hour, 2 hours, 3 hours later. And then the person comes back a week later on the same day, same time. We ask them to have the same meal the day before, to try to have everything as the same as before. And now we give them the other, either the plant or the placebo. And then we do the same work. And then we subtract the person's circadian cycle, because we have a circadian cycle. The number of stem cells is not constant. It peaks at about 5 a.m. It kind of goes down throughout the day. So we need to subtract that from the response with the plant. So that's kind of the work that leads us to document what is the effect of a plant on stem cells.
Incredible. So what did you see when you started to play around with these different compounds?
So we took this AFA and we started to test it, and very quickly we found that the product was putting more stem cells in circulation. At first, actually, I thought, because we had already documented that the polysaccharide in AFA had an effect on NK cell, and the effect was to trigger their migration. So we started with the idea, and that's what I mean when I say scientific investigation. You have a hypothesis, you're proven false, but you see something else and you take a different path. I thought maybe the polysaccharide does the same thing to stem cells, trigger the migration into tissues. So as you make stem cells migrate into tissues, then they can repair. And that's how you stimulate the repair of various organs. And what we saw was not a decrease of stem cells like we saw in NK cell, but an increase. So we thought maybe there's a mistake. We repeated, and then it became clear very quickly the product is mobilizing stem cells. So we thought maybe it's the polysaccharide that is doing the same thing. So we extracted the polysaccharide. When we give the polysaccharide to people, to ourselves, we see a decrease in the number of stem cells. So blue-green algae actually has two active compounds. One that triggers their mobilization from the bone marrow into the blood, and the other one, their migration into tissues, just like NK cells.
Yeah.
So that's how it started. But at first, when you document something like this, the response from the scientific community, and it's not a criticism, it's sort of a healthy response. It's well, if you don't have a mechanism of action, what you're documenting may be just random. Like, it may not mean anything. And putting more stem cells in circulation may not mean anything for health. If anything, maybe it's bad for you. So how do you now answer these questions? So we started to study mechanism of action, and we documented the mechanism of action of the active compounds. We isolated the active compounds. We did proof of concept in animal models of injury to document the enhancement of healing or recovery, and so on. And so that's kind of how it was all developed.